In this brief movie, I want to talk about how phylogenies can be used for many different things and how they can show up everywhere within biological literature. Phylogenies are used in lots of different biological and medical disciplines, most commonly ecology, evolutionary biology, but all paleontology and anthropology, but also they frequently show up in bioinformatics, computational biology, also epidemiology, and even cancer biology. One thing that's important to know is that any taxa or biological entity can end up on a phylogenetic tree. Often it's species or subspecies, maybe populations, but any taxa or genera, we've seen families or even orders can show up, but it could also be much smaller scale. It could be patients, individual organisms, tumors, even individual cells can show up on a phylogenetic tree. So a classic example of individual patient showing up is a phylogenetic tree that was used to trace infections that were thought to have originated from a dentist that infected his patients. So on this tree, each tip is a person. It's either the dentist or patients. There's patients A, B, C, D, E, F, and G. And putting these patients along with a number of controls onto the tree were used to infer the pattern of disease transmission. The patients in this case are patients who were the patients of the dentist. Blood samples were taken and HIV extracted, HIV DNA extracted, rather HIV RNA extracted, sequenced in order to then characterize how similar the different patients, the strain population in each patient was. Then also a number of controls were sequenced. These are other patients from that had no contact with the dentist, but also were HIV positive. Genetic viral genomes were sequenced from each one of those, and then the sequences compared between the patients of the dentist and the control groups. And something that shows up quite plainly is a clade where all of, or most of the patients, few outliers, but most of the patients cluster together as a clade, and that clade also includes the dentist. So patients C, A, G, B, E, all are within a clade, and they show up within the dentist. And most of the controls, rather all of the controls, a few of the patients, but all of the controls are outside of this one clade. So the patients are in a clade with the dentist. This is strong evidence that the patients got their infection from the dentist. What's interesting is there are two patients here that are outside. This could be these are patients that picked up HIV somewhere else. Another interesting feature is that the dentist shows up in two places and patient A shows up in two places. So think for a second how that could occur. There's two possibilities. We'll focus on patient A. One possibility is that patient A had been infected twice with HIV. Another option is that HIV evolved and developed into two separate populations within them. Patient A, both the patient's A samples occurred within the same clade. So I haven't read up on this, but it's possible that patient A had evolution occurred within patient A, and perhaps this, for example, this strain here evolved separately from this common ancestor back here. The dentist has two separate strains, so it's possible the dentist had this evolution phenomenon, or the dentist was infected twice. For example, the dentist is out here, so the common ancestor of this clade is right here, so that means all of these strains, the evidence indicates that they descended here. So this could be the ancestor of all of the strains lies here. It's possible the dentist picked up a different infection and that is indicated by this strain here, or that the strain evolved, and then the common ancestor of this clade is here, and then this is the new strain that evolved within the dentist. So this is very topical. Currently, phylogenies are being built for COVID-19. Early in the transmission process, individual patients were being sequenced in order to understand whether the virus was being introduced multiple times, whether it was coming from the East Coast or the West Coast, trying to understand how a virus that we know originated in Asia, where it traces back through. Tumors can also be placed on trees. There's some interesting applications of phylogenetics into cancer biology. Cancer spreads and grows over time. So in this case, this study looked at a large tumor. They sampled cells from the side of the tumor, and they could then trace by sampling through different places throughout the tumor, they could then trace how the tumor was changing over time. So they found that on the edge of the tumor, there appeared to be the ancestral type of cells. In the interior of the tumor, there was a different group of cells. There appears to have been a split here and a clade of other parts of the tumor formed over here. So there's the founding clone. There was an initial split and then another split here. So this is one set of descendant cells. Here's the split down here with another cells over here on this side that are descended from this group here, and then further continued split. So you can actually trace the set of mutations and chromosomal aberrations throughout the cell, physically tracing how as the tumor grew, mutations happened within the cells. Similarly, cancer spreads throughout the body. Individual tumors don't just grow, but also cells can become released and spread throughout the body. So you can actually compare hypotheses for how the process occurred. In this study, they were looking at whether ovarian cancer that had metastasized and spread to different parts of the body, whether the same ovarian tumor had cells had come off of it multiple times and spread to different parts of the body. So a single point source of origin spreading to different spots, or whether there was kind of a chain where an initial malignant cells initially spread to one part of the body and then they split again. And actually, in this case, this says tree topology. This indicates that most of the support was for this form of spread, where first the initial tumor was in the ovary, it then spread to one point in the body, and then it spread to 2 points from there.